Hello, this is Alex Lamb. Today I have Nathan Killoran from the University of Toronto with me, and we're going to talk about his recent work on modeling DNA with deep generative models. So to start things off, can you explain what motivated you to start considering working on DNA with deep learning and generative models in particular?
Sure. I just wanted to say right off the bat, thanks for having me. It's great to be on with you, Alex, and chatting today. In regards to your question, I was working with my colleagues at U of T. That's David Duveneau, Brendan Frey, Leo Lee, and Andrew DeLong, and we were kind of trying to find a topic that would work for exploration. And this is with generative models. He's done lots of work on generative models for chemistry, making chemicals from generative models. And Brendan's group has been working for a number of years in applying, deep learning to genomics data. So it was a very natural kind of proposal to just combine generative models with genomics and DNA. So really the motivation, I would say, is just scientific curiosity. We had some interests and we started asking questions, and we discovered that actually those questions are probably worth pursuing, at least as a kind of exploratory work.
I see. Is there a reason to think that DNA itself would have an interesting statistical structure? So my intuition for this is that because the DNA is sort of passed through this complicated function to form the organism, it seems like simple differences in DNA could lead to complicated variations in phenotype, which could be analogous to the way that a latent space for a generative model can be something simple like marginally independent Gaussian, and the variations in phenotype or the variations in the output can still be fairly complicated.
I would say probably the first point is that, from my understanding, that independent Gaussians is maybe a bit too simple. It's quite a lot of complexity there in the genetics data. There's actually quite a lot of interaction between which makes the statistical structure more interesting. There's lots of ways that you only have a particular disease if you have multiple mutations in different spots rather than just a single mutation. There are single gene mutations that cause disease, but they're not all like that. There's between different.
Do you think that there's some kind of general difference in the statistical properties of like unhealthy variations that are associated with disease versus like just normal differences in healthy people?
I'm not so sure. I don't have a great answer for that. I would say that the interesting thing is that most people, you hear these statistics, we share like 99.9% of DNA with each other. So
yeah,
the statistical variations in DNA are quite small within the same organism, the same species.
Yeah.
And it's still that small amount of variation leads to quite different exterior phenotypes or different, susceptibility to diseases. So even though a lot of the DNA is conserved, there's not allowed to be any changes there. There's still quite a lot of interesting statistical structure on a small subset that is allowed to be changed.
I see. Do you think that maybe the long-term goal of this field of generative models with DNA is to understand the conditional distribution, which maps from the genotype to phenotype? And do you think that there will be a way to learn this conditional distribution in a way that it generalizes off of the distribution of genotypes that naturally occur in organisms?
Sure. Maybe I answer in two parts. So the first part was about the statistical. Say again, sorry.
Yeah, the first thing was like if the long-term goal of the field is to model this conditional distribution.
So I would say definitely it would be a great long-term goal to be able to say, if you have this sequence in your DNA, or this mutation here, this leads to this phenotype, this characteristic, this susceptibility of disease. And to be able to do that, across the whole genome, I would say that's maybe a bit ambitious, though. There's lost along the way between the microscopic DNA sequence and the macroscopic or other organism. And so it makes probably more sense to find some conceptual plateaus, some stopping grounds along the way between those two steps where, you want to be able to explain not just from genotype to phenotype, but genotype to some intermediate level, then to phenotype. So for instance, Brendan's group has been proposing for quite a while now this notion of a molecular phenotype.
Yeah.
And there the idea is, well, let's take the DNA and just try to predict what kind of molecules are going to be floating around in the cell. So what are the different RNA transcripts, the different proteins, the other biomolecules floating around the cell, conditioned on what the person's or what the cell's DNA looks like.
Would you say that the long-term goal of this field is to understand the conditional distribution, which maps from genotype to phenotype?
I would say that's definitely a major kind of ideal goal we would aim for. If you could tell someone, based on your DNA, I know you're going to have this appearance or this characteristic or this extra risk of a disease. If you could do that across the entire genome, then that would be fantastic. It's a bit of too ideal a goal. It makes probably more sense to find some kind of conceptual middle ground between the microscopic genotype and the macroscopic phenotype.
Yeah.
Stopping ground where you want to predict not just based on your DNA, here's your phenotype, but here's some sort of intermediate representation for things, and it's not coarse graining all the information.
Yeah. What would be an example of one of those like intermediate type goals that you'd want to model?
Since a natural unit is the cell, it's a small unit. Its structure is kind of based about what the DNA says. So the DNA contains instructions for creating RNA transcripts, for creating proteins and other biomolecules that end up kind of floating around in the trations. So Brendan's group in particular has been pushing for a while, and in the field as a whole has been moving towards this notion of kind of a cellular phenotype.
Yeah. I see. Is it generally possible to like measure these things going on in the cell?
Yeah, I mean, there's lots of different types of things floating in the cell. So there's RNA transcripts, there's different proteins, and for sure we can measure some of these things.
I see. Do you think that it'll be possible to learn this conditional distribution in such a way that it generalizes off of the distribution of genotypes that occur naturally?
Absolutely. I think that's actually one of our goals with this work. One of our motivations was to say biology is kind of lazy, so it finds a good solution to get, to solve a problem, but then it doesn't try any harder.
Yeah,
it's energy inefficient maybe to try it, to search for more solutions. So we thought there might be some more room above where the biology stops. There's certain things that have to be respected, certain characteristics that have to be respected, but maybe we can push that further than the biology would take it, and we can kind of, see what are the extremes of, say, expression of a particular gene or binding affinity.
Right. Do you think that there's any possibility of sort of genetically engineering an organism to have actual DNA changes from a neural network? And if so, how far away do you think we might be from that becoming real?
Yeah, so that kind of builds upon the previous question. I definitely think that it's a natural way to go. I mean, people are looking into bioengineering, genetic engineering. I mean, even on the level of microbes, right? So you don't have to necessarily talk about bit larger species like humans. You can talk about microbes, and that's also tremendously valuable. Microbes are used in industry for creating pharmaceuticals, creating fuels, industrial solvents, food production. So being able to say change the genome in a microorganism so that it produces 10% more biofuel or something like that. I mean, that would be fantastic if we could do something like that. And that's even just say maybe 12 million or so nucleotide genome versus human DNA, which is 3 billion.
I remember when I was doing my undergrad at Hopkins, there were a number of people who were working on the yeast genome. And I guess they picked yeast because I think it's the simplest eukaryotic organism, but they were trying to identify like a minimal genome that was necessary for the yeast fitness so that they could use the other slots to engineer things into those places.
Yeah. So actually, the yeast genome, I believe people have been able to synthesize the entire yeast genome artificially.
Yeah.
So that tells us the scale of the techniques right now. We're not necessarily at human level, but for sure we can synthesize quite long sequences, quite long genomes.
Yeah. I see. Moving more to like machine learning stuff, do you think that the size of DNA, 4 billion base pairs for a human, will present a fundamental problem for machine learning? Do you think that research is going to end up focusing on understanding small segments of the genome? Or do you think we'll have work that tries to understand the joint distribution, either just over genotype or also with phenotype for an organism's entire genome?
I would say that's so certainly 4 billion characters in a data set. That's a lot, even for kind of modern machine learning methods. But conceptually, I think that length is not necessarily the biggest problem. There's lots of other things that are going on that might be more complicated than just pure length, pure size of the genome. So, for instance, there's lots of interactions between different genes. You might have a particular susceptibility to disease if multiple genes are active or present. So those interactions are quite important. There's also lots of extra structure on DNA. It's not just a single, double-stranded helix, but it's got some 3D structure. So DNA is wound in the cell. And depending on the conditions or the kind of context of its environment, the certain sections of the DNA will unwind.
I see.
So there's this 3D structure.
Is it generally possible to just measure what this? I guess, you're saying it would depend on the environment. Is there a way to kind of know what that 3D structure might look like, or is that pretty hard?
So for sure, we know the kind of general. So we know that what happens is you have these histones. And the DNA is kind of wound around these balls. And then it's packed away quite tightly, like you wind a thread around a spool. So you wind around these balls, and so that's how you can pack such a long sequence, a long biomolecule into a very small cell. Is because it's wound very, very tightly. So most of the time the DNA is packed away and not being used. So there's lots of kind of extra structures that are just not present when you look at the raw sequence data, the raw base pairs. And I think that will pose a kind of bigger obstacle than some of the other things like length.
Would you say the packing, it's mostly like a fixed structure, or does it kind of change a lot depending on the environment?
I think there's kind of two phases. So there's basically two stages or two phases. Usually the DNA is wound quite tightly. And then there's a stage where certain parts are unspooled, and those parts are used as kind of, to read certain sections of the genome.
Yeah.
transcribe into proteins. And you don't always need to make proteins. So depending on your developmental stage of the cell, for instance, you don't need certain proteins. But the DNA contains all the instructions on how to do all those things at all the different stages.
Mm-hmm.
So it's maybe like a book, and you just need to look at, one page at a time kind of thing.
I see. That's interesting. I suppose that ties in pretty nicely to my next question, which is if you could kind of describe the high-level structure of DNA with some focus on what kinds of statistical dependence properties it has. Like as you said, it probably doesn't make sense to think of it as one long discrete sequence, but maybe if you can think about the groups that tend to get unspooled together, maybe that's kind of a chunk that could be thought of as an example in a machine learning sense.
So I guess kind of the very simplest chunking of DNA that you could do is you could break into chromosomes, right? So chromosomes are actually like physically independent molecules that code different parts of the genetic code. There is a little bit of interaction between them, but mostly they're quite independent from each other.
I see.
So conceptually, that's probably the biggest kind of chunking you can do. And actually, one of the experiments I did in our paper was I just concentrated on a single chromosome rather than the whole genome.
Maybe this is a tricky question, but do you know why DNA is divided into multiple chromosomes instead of like one giant chromosome? Like, does it have a functional purpose, or is it just like a historical kind of thing?
To be honest, I'm going to say I don't have a good answer to that question. There probably is one, but I don't have it. It's not my specialty.
I see.
So in terms of DNA, I said that chromosomes are the biggest kind of hierarchical structuring you can make with the DNA. But then kind of the next conceptual step is to look between coding sequences and non-coding sequences.
Yeah.
And something like 90+% of a chromosome will actually be of this non-coding type.
Right.
And what's the difference between coding and non-coding? Well, coding is like the instructions to build a particular protein molecule.
Right.
And the non-coding is everything else.
I forgot who it was, but there was one person who said that one way to think about this coding, non-coding thing is to imagine looking at a computer program and all the lines that are print lines are like coding sequences and everything else is like a non-coding sequence.
Yeah. I like to make the analogy like maybe the non-coding sequences are just like white space in a text. If you cram everything together, it's harder to find the individual words, but if you separate them, then it's easier to compartmentalize them. But there's actually the non-coding sections, maybe it doesn't code towards a particular protein, but there's still potentially some functional behavior there. So surrounding the coding sequences, there's typically upstream and downstream some extra structures that help with like navigating or finding that sequence. So certain patterns that a protein can bind to and discover where the nearby sequences are. So there's always like structures hidden within the kind of apparently unstructured parts. But there's also the potential that the unstructured parts, some of it is just like genetic fossils from previous stages of evolution that just never got cut out.
Yeah.
They're not expressed anymore.
Yeah. That makes sense.
I should say there's probably one extra kind of hierarchical step we can take. So when I talk about a coding sequence, this is like a simple kind of recipe for making a protein molecule. But these actually aren't continuous themselves. They're broken up into what's called exons and introns.
Yeah.
So the exons are the parts that actually end up having code words in them.
Yeah.
And then the introns are just kind of like spacers in between those. And you might have, alternating pattern exon, intron.
Yeah.
The introns eventually get removed and the exons get stitched together. And you think, why would we have this extra layer of complexity? Why don't we just put the entire coding sequence all in one spot?
Yeah.
What this allows for is it allows you to kind of take these fragments, these exonic fragments, strip out the kind of white space in between them, and then you can think about combining them in different ways.
Yeah.
So depending on the molecular conditions of the cell, you might not need that middle piece. So you just combine the two end pieces together, and that creates a slightly different protein than the one you would have created under some other condition. So it gives an extra layer of complexity to the DNA. So there's actually quite a lot of stuff going on there that's, depending on the level of kind of abstraction you're looking at, has more and more structure to it.
I see. Why did you use a GAN architecture for your generative modeling? Do you feel like another generative model could work, or is there something special to GANs?
So actually, the GAN was one of the last things we tried.
Yeah.
And the reason we ended up sticking with the GAN is it actually ended up, the one we did was this Wasserstein GAN,
Yeah.
which is a kind of a second generation GAN, I would say. And
Yeah.
the reason we stuck with that one is because it seemed to work quite well. It was really, really easy to train. There was very little need for hyperparameter tuning or anything like that. So it was very robust and very flexible. And we wanted to do lots of different experiments on different data sets. And we wanted to kind of combine a generative model with a predictive model and differentiate through the whole structure. So the GAN kind of, the reason we chose it was mostly just because it ended up kind of working well. We did try a number of other generative models. So I tried a recurrent neural network, variational autoencoder, and a variant of the pixel CNN architecture.
Yeah. But I guess with a 1D convolution.
That's right. So I mean, a pixel CNN is basically like a recurrent network where you're allowed to look over the kind of the whole past history with a ConvNet.
Yeah.
And it's used at Google DeepMind for speech recognition, all sorts of really interesting things. And that one I would say actually did work. That was pretty good at modeling, but I found it just a little bit unwieldy, a bit kind of bulky for my needs.
I see. Oh, right, because sampling from it is a little bit tricky.
Yeah, especially we wanted to be able to do kind of design, not just sampling from the generative model, but kind of push it towards interesting new areas. And in the end, again, it became very flexible, and the other one was a little bit less flexible for that.
Right, because you can just express it as this kind of simple sampling function.
Yeah.
I see. So a lot of times when GANs have been trained, you can look at the latent space and you can find certain kinds of interesting structures, like walking along the manifold and seeing like gradual changes, or doing interpolation, or kind of doing the simple like addition, subtraction on the manifold. Was there anything interesting you found in the latent space of your learned model?
That's a interesting point. So we actually did find some cool structures there. I did kind of look for them, because this is kind of standard in GAN papers or generative model papers, is to look for these kind of simple structures.
Yeah.
I don't know if it really explains much, but at least shows that your model is learning some interesting structures.
Yeah.
And so one thing we discovered is I trained a model on a single chromosome. So that's the biggest chromosome in human genome. I took all of the DNA from that, and I kind of fed it into a GAN, and it was supposed to learn to kind of duplicate or generate sequences that looked like it came from this chromosome.
One sec. So you chunked the chromosome into a bunch of examples, right?
That's right. Yeah. So actually, you can, if you just download the kind of stock file from the genome browser, it comes with sequences that are 50 characters long.
I see.
So a new line, every new line has 50 characters. So it was already kind of pre-chunked for me.
I see.
But it's an arbitrary splitting. Like it doesn't mean there's a functional split at those numbers, but it was just convenient.
Yeah.
So we trained on this quite large genome chromosome data, and I looked at the latent space. And one thing I discovered is that it kind of picked up the idea of DNA complementation.
Yeah.
So DNA complementation, DNA is a double strand. So there's on one strand, there's these 4 characters A, C, G, or T, and then the other strand can kind of join up with it by having the complementary sequence. So A binds with T, and C binds with G.
Yeah.
And so those guys always come in pairs, but that's always a pair on a double strand. And we actually trained the model on a single strand, and we still discovered that it picked up this notion of complementation from the single strand sequences.
Right. So you have the single strand, but then like the other strand would presumably be somewhere else in your data set. It's just not explicit.
It wouldn't be, no.
Wait, but if you had the whole chromosome?
It was just one side of the chromosome.
Oh, okay.
Just to make the experiment clean, right? So we just took the one side, the one strand, the entire length of that strand.
Yeah.
So actually, I was talking to a colleague of mine last week, and he said, "So I read your paper, and how did your model learn that? Like, it's not really at all clear how the model picked that up, because, as I just said, we only trained on the one strand. So I guess there's some sort of like hidden statistical structure in there that reveals the presence of this complementation somehow. But I don't know what it is. It's just cool that it picked that up."
Yeah. That's not obvious to me either.
Yeah.
What would cause that? If I had to guess, though, maybe there's a certain type of like function that can occur in that DNA and in the complement as well. So it's just easier for it to use sort of common features for both of them, and then
I mean, one hypothesis, maybe quite simple, is certain nucleotides might occur in specific concentrations, and you might expect the concentration of A and T to be the same, even on a single strand. So maybe that explains it. But we thought it was pretty cool to find this kind of thing.
Yeah. So one thing that's interesting is, like with most generative model papers, most people will kind of start with MNIST, and then they kind of have this notion of a hierarchy of data sets of increasing difficulty. Like you do MNIST, and if it works on MNIST, you move to SVHN, and then if it works, you move to CFAR. Do you think if someone's doing like generative modeling for DNA, like this thing where you take a single strand of the chromosome, you do these 50 character chunks, do you think that's going to kind of be like MNIST for this domain? Like it's the natural starting point, or could it be something else that's be better?
Yeah, I mean, definitely it could be. Maybe this will start a trend and it will become the MNIST for DNA. But there's lots of interesting starting points, and I think you're going to see different things depending on what kind of data you put in there.
Yeah.
So you don't want to just look at one type of data, because that's not going to reveal the full picture. So especially in this field, it's important to gather data from multiple different sources and kind of combine them together to get the best understanding. I don't think you're going to have just one, maybe as like a toy task, like a first test, a sanity check, you would check chromosome 1, right?
Yeah. So a lot of the DNA research, at least that I've seen, and this would be more like in the social sciences, uses linear regression or even simpler models in the space of these single nucleotide polymorphisms, or SNPs. Do you think that deep learning approaches on raw base pairs are just going to supersede this SNP representation, or do you have a view on that?
Yeah, so maybe I just quickly overview what this single nucleotide polymorphism means.
That would be good.
Because I find there's a lot of jargon in the field, so it's good to understand that. So you can imagine just doing taking a single gene, say, and saying this gene is found in all humans, but at the fifth character, the fifth nucleotide, there's some variation amongst the population.
Right.
So that's something that's allowed. It doesn't prevent us from becoming fully formed organisms. So, can we see if those things are correlated with some characteristics that we observe in the population? So we take, the input data is the different variants that the different people have.
Yeah.
And the kind of thing we want to predict is some phenotype or some susceptibility to disease. And you want to say, like, let's just look for, simple statistical correlations between those two things.
Yeah.
So when you're taking like a genetic test and they say, oh, you have a slightly higher risk of Alzheimer's, well, you might be
Yeah.
doing something like that under the hood.
Yeah.
So I would say that deep learning models, as we've seen in other fields, they should definitely, if you have features that you're trying to learn, they
Yeah.
should definitely help. We've seen in other fields that these things, can only increase the understanding or the modeling capacity we have of our systems.
Right.
So it's important to kind of, I don't think it'll necessarily supersede it, but it'll kind of like coexist with it and provide a better perspective or an alternative perspective.
Right. Uh, to me, it's interesting because the whole SNP thing, when I first heard about it, reminded me a lot of these bag of words models from NLP.
Okay. Yeah. Yeah.
Yeah.
But I think yeah, it's just like maybe your first approximation. That's the simplest model you can come up with, your simplest representation. But maybe there's more complicated features than that, right? So let's push it a bit further.
Yeah. So taking things in a somewhat different direction, do you think that the statistical structure in DNA will have connections to sexual reproduction? For example, the simplest such property is that sexual reproduction might encourage traits to be expressed across many different genes rather than just an interaction between two specific genes, because such an interaction is likely to be destroyed during sexual reproduction.
Um, yeah,
I mean,
I don't know if I don't know if I have a
yeah,
great answer for this one. I would say that for sure there's some during sexual reproduction, your
yeah,
mother and your father, they have
yeah,
their own chromosomes, and those chromosomes will kind of cross over. So you
right,
might your kind of your chromosomes that you inherit,
yeah,
part comes from your father, part comes from your mother. Um, not all parts are kind of mixed. Like certain things between the mother and father are completely the same.
yeah,
There's only small variations on that, and I would say that it is important to consider interactions between multiple genes or multiple kind of spots on the genome. But a lot of the time, when genes cross over between the parents, sequences that are nearby to each other tend to cross over together. So it's not
yeah.
like a complete, throw everything in a bag and pull it out at random. Like they kind of go with structure. They're not just willy nilly mixed
I see.
together.
So it's not directly related, but one thing that's kind of interesting is with bees. Apparently, the male develops from an unfertilized egg, and then females develop from the fertilized egg.
Okay.
I don't know. That's so males only have half the DNA. I don't think there's anything super interesting to get from that, but I do think
Uh,
I do think it's cool.
but we know for sure if there are kind of deficiencies, if you fail to inherit a chromosome from your parents, you can cause different diseases. So we have to
Yeah,
be um, there is definitely you want to there is a role for sexual reproduction. You want to recognize that, but
yeah,
I don't know if it's in terms of the other things we've been discussing. I don't think it breaks the statistical structures of the DNA too much.
right. Um, do you think that deep learning methods could be practically used in these gene-wide association studies? For example, like a deep genome-wide association study?
For sure, yeah. I mean, so we can kind of think of these genome-wide association studies as the kind of um, a large scale, what we talked about these SNP, single nucleotide polymorphisms
Yeah,
earlier. So you kind of think of that as looking across the entire genome, looking how people's genomes differ, and associating that statistically with um, some phenotype or some susceptibility to disease. And the models might be quite simple in the past, but for sure we've developed
yeah,
all sorts of new probabilistic modeling techniques and learnable parameters in neural networks and implicit densities and things like this. So there's definitely room for these kind of things to play a role and to increase our understanding in these genome-wide association studies. And actually, there's a interesting work last year in October from Dustin Tran and David Bly doing exactly this, looking at genome-wide association studies using kind of modern probabilistic techniques and finding you can get quite a good improvements in the power.
Oh, interesting.
So I would say there's probably the doors are just opening right now. There's lots of opportunities to do more stuff like that.
What would you say was the most surprising thing that you found out about DNA when you started working with that?
For me, I actually, I would say something called the epigenome.
Yeah.
So the epigenome is kind of like you have your genome, it's all your DNA. And as I said, it's kind of coiled up in the cell. And then the epigenome is kind of like these markers that sit on top of that structure. And they kind of, they'll enhance or suppress certain things. They'll kind of expose certain sections of the DNA or hide other sections depending on, maybe environmental conditions or something like this. And the crazy thing about that is that it's inherited. So you're not just inheriting your DNA from your parents, but you're inheriting this epigenome as well, which is these markers that are sitting on top of your DNA.
Wait, only from your mother, or?
That's right, yeah.
Okay.
Yeah, so the analogy I like to make is like Harry Potter. So Harry Potter, he goes to Hogwarts and he takes this class on potions, right?
Yeah.
And they have a textbook that says, every page is like a recipe for how to make a particular potion. That's like the DNA, right? And in one of the books, Harry, in his potions class, he gets the textbook from his instructor that's been kind of appended, right? There's all sorts of notes written in the margin, and it says extra things about how to make them more potent or how to swap out ingredients. So that's kind of like what the epigenome is. You're inheriting from your parents this kind of appended recipe book.
Yeah.
I found that really, really fascinating. It's not something that I kind of ever picked up in high school level biology courses.
I see. So one thing is, if you had some metric for like what the genes are doing, like it could be like an end-to-end phenotype or something else. Maybe if you regressed like the father's gene against the trait and the mother's gene against the trait, if they're kind of equally correlated, maybe that means that it's not epigenetics. And if it's more with the mother, maybe it's more likely to be epigenetics or some other kind of environment. Or at least maybe you could kind of bound the impact that way.
Yeah, you could think about that. I mean, I don't have a good response to that. I don't know too much about the epigenome.
Yeah,
but it amazed me that it was there.
I see. So a lot of people have been hearing about CRISPR. Could you like briefly explain what that is?
Sure. So CRISPR is it's actually originally a kind of mechanism that's bacteria or microorganisms had to let's say excise when a virus was trying to implant its own genetic material in the microbe.
I see.
So you need a way to kind of like recognize that foreign DNA that's been injected and kind of splice it back out, right?
Yeah.
And so that's the kind of basic principle. It exists in nature, and then we kind of in the last 5 years or so kind of engineered this process to be a way to write the genome. So you take this mechanism, this CRISPR, let's call it an apparatus, and you use it to kind of make changes to the genome with single nucleotide resolution. So you can really think about going into a cell and on the fly swapping out nucleotides for other nucleotides as you like.
Yeah. I see. And it's I guess it's like a pretty general method. Like you can mostly replace things you want to?
Yeah, I mean, there's still lots of studies ongoing with these things, but it's become a very big research area because it's quite a powerful tool and it's actually quite inexpensive to use. And so, I mean, it's become the kind of de facto standard for these kind of ideas of changing, writing to the genome.
I see. What do you see as the interaction between deep learning and CRISPR?
I would say that, so as I said, CRISPR is a way for writing to the genome, but we need to know a way to decide what to write. You know, we need to be able to understand what are plausible changes to the genome, what are changes that are going to have a desired effect.
Right.
And that's the role I see for, you know, machine learning models, is to predict the changes that are necessary, fill in the spots that are, you know, they have to be plausible, but they don't necessarily have to be exactly understood.
Yeah.
So those models will tell us what to write to the genome, and then CRISPR will give us the mechanism for actually writing to genomes.
I see. I wonder how long it would take until you actually have an organism where some of its DNA was selected or designed by a deep learning model.
Yeah, I would say that for sure it won't be very hard to synthesize DNA kind of in a wet lab. That has been proposed by a deep learning model. And as you said, if we combine this with CRISPR, then we can put it in an actual cell.
Yeah.
I mean, I would say that the tools are all there right now. It's just a matter of like how much you trust the tools to do what you want them to do, right? That I think is a bigger issue than the mechanism for actually. People are already using CRISPR to put changes in DNA, and so the deep learning model is just a way to suggest new ones, right? So it's just a matter of trusting the model, building the models up enough that we trust that they're going to do what we want them to do.
Right. So this segues nicely into in one of your experiments, you train a classifier to predict a protein binding, and then you train a generator to produce sequences with a high binding score under your classifier. Mm-hmm. Could you talk about that a little bit?
Sure. So the idea here was basically we take a generative model, and the generative model's purpose is to kind of provide us a latent space where DNA is encoded in a continuous way.
Yeah.
So it's kind of from these coded continuous latent codes, you pass them through the generative network, and it produces a DNA sequence.
Right.
And it should be able to not only learn to produce sequences, but to learn to produce plausible sequences given the data set that we trained it on. So you can think of it as a kind of prior on the set of natural DNA, or the set of DNA that's relevant to the problem. And then kind of as a separate thing, you train a predictive model whose job it is to take in DNA sequences and predict some particular property. We studied binding, for instance, which is a relatively simple property. And so you have this predictive model and you have this generative model, and they can be trained kind of separately. And then you both them together. And you then do a kind of a back propagation through the entire network, the joint network, and you have you're able to move around in the latent space to positions that will lead to, you know, higher prediction values or higher scores.
Right, and you were able to produce values under that classifier, which are higher than you get for any real examples. Actually,
that's right. That's right.
Considerably higher.
Yeah. So I mean, I would say that maybe the structures we considered were quite simple.
Right.
But we did do an experiment where we took a kind of range of allowed values for the predictor.
Yeah.
We cut out all the sequences of high value. So we know that there are sequences of high value, but we cut them out and we trained on that truncated data set. And this process that we used was able to kind of push back into that region that was cut out just from the smaller truncated region. So it gave us some hope.
I see. So you're saying that you took your original data, you cut out the parts that you knew had high value, like, for example, the high value examples, and then you're saying you train your process on these low value examples, and then you found that it had like a high subsequence overlap with the high value examples.
I wouldn't say that the sequences were the same that was generated. And in fact, we did a test to see whether the generated sequences and somehow the test data were similar.
Yeah.
And they were different enough that we were confident it wasn't just, reproducing those guys. But it was an experiment that allowed us to we wanted to push past the kind of the training data, right? But we knew that there was room there to move because we kind of artificially created the situation. So it gave us some confidence that if there is some structure in biology that's kind of beyond where natural DNA stops, but there's still room there, that we could actually push into that area.
Right. So one thing that I guess concerns me a little bit is there's this work on adversarial examples with neural nets. So are you somewhat concerned that you could be like finding kind of like adversarial examples on the classifier, or you could be finding regions where it kind of isn't very accurate?
So that was definitely a concern. We tried to structure our experiments in a way that we could still validate that sequences were being created that weren't adversarial, that still had the right property, wasn't just an artifact. But I should say that biology is a experimental science. You can actually just go and do some wet lab work. So we were proposing sequences. In principle, we were using a predictive model to predict the values of those sequences, but we can also measure the values, the binding affinities of those sequences, and verify in the wet lab that this is actually a real phenomenon. So that's one way to rule out adversarial effects, is you can go and actually just test. And you can even envision kind of an active learning situation where you propose a bunch of sequences, you test those, and it gives you, it tells you which ones you were completely wrong about. Maybe those are the adversarial examples, and the ones you got right. And you take that data and you retrain with that new data, and you can kind of improve your model this way, make it robust to adversarial examples that way.
I see.
And in some sense, we were actually we were looking for adversarial examples. We wanted things where a small change in the DNA led to a huge change in the prediction.
Well, so I guess there's two senses. One is like for the real process that leads to the binding. It's like you want to find adversarial examples for that. You don't want to find things that are just fooling your classifier or just making
artifacts of your data set or something, or your training method. That's right. So I mean, we were searching for things that were kind of extreme, but of course we want to still make sure that they're not adversarial in the sense of they're spurious, right? They don't actually reflect the data. One advantage is that working with discrete data like DNA, it's clear to see an adversarial example. If I flip a single nucleotide, then a human can spot that difference, right? Whereas in adversarial examples in images, you can imperceptibly change the values of pixels, so the human observer can't detect it, yet the model is fooled by it. So at least for discrete data, we're able to kind of validate with the human eye a little bit easier when you've done an adversarial change.
Yeah, I see. So one thing is that you used sort of a deep dream or activation maximization technique as a tool to design new DNA sequences. What motivated you to do that?
I would say that the deep dream, the idea behind deep dream is, say I have a neural network who looks at images and then makes predictions about a class of an image. So it has images of dogs, and then you predict whether it's a cat or a dog or something else, right? So you have a continuous input and a discrete output. What we were doing is kind of the flip of that. We were taking a discrete output and then having a discrete input and a continuous output. So, and then you say, well, deep dream method is something like running a network in reverse. So I look at I start maybe even with random noise at the input, and then I look at the activation of the class I'm interested in, and I follow some gradient until I move around the image. I reshape the image to more strongly activate the dog class. Right. And so we wanted to do the same thing as a design tool for what we were doing. So start with a sequence, predict its properties, and then calculate a gradient of that that property with respect to the input sequence and find ways to kind of improve it. Right. So this deep dream is kind of in the popular literature. It's it's kind of a fun thing. It gives these nightmarish images, but it's actually quite an interesting method for design. We were interested in design.
Yeah,
and if you combine a generative model with this design tool, you actually get really interesting powers.
Yeah, I mean, one thing that's interesting is if you do this deep dream procedure, it can move you off of the observed data manifold. Yeah. But if you do gradient descent through your generator, it should more or less stay on the data manifold.
That's exactly right. So the generator serves as a kind of prior to the plausible sequences. And you can kind of trade off between how close you stick to the generator's prior and how much novelty your generated sequences have. So there's a bit of an art there, but I guess that's an element of creativity. How much do you want to extrapolate versus how much do you want to play it safe, right?
Yeah.
But in principle, it allows for that quite flexibly.
Yeah. So do you think the method for doing deep dream, this activation maximization method, could generalize and work on other discrete sequence data like natural language?
I would say in the long run, definitely.
Yeah.
We actually, I would say, still need better kind of neural network models for natural language, generative models for natural language than we currently have. I would say that the ones we have now, they approximate natural language, but it's still clear that it's not real natural language.
Right.
Especially with long-term correlations, they're just not modeled very well. But definitely, I think that the techniques should be able to apply. We saw here that they worked for DNA, which there's only four letters in the alphabet, say, so it was a bit easier.
Yeah. One thing I do think is interesting is if you look at how many pixels you have in like an ImageNet image, it's I think it's like 45,000.
Mm-hmm.
And I believe that's about the same number of words that you would have in a short book.
Okay.
So it could be possible that we're not used to thinking of as long-term as long of dependencies as we potentially could be with that.
So actually, it's an interesting story. I said that we use this Wasserstein GAN as our kind of favorite model for generating DNA. The first place I encountered that was actually in a paper. They were using this GAN for natural language processing as a generative model for text.
Yeah.
So I pretty much just in my first experiments copied that same architecture that was designed for English generation, and said, well, if it can do English with 26 letters, let's try DNA with 4 letters, and it turns out it worked pretty well.
Yeah. So actually, that makes me think of another thing. One big challenge with generative models research in general is how you evaluate the quality of your generative model. So a lot of people will do they'll just do visual analysis of samples, which I guess probably is hard for DNA. But in terms of quantitative evaluation metrics, there's this inception score. There's not too much that's good. What do you think could be good ways of evaluating the quality of a generative model on DNA? And is there anything that you found was effective for analyzing?
Yeah, so we did have this concern, and one thing we tried was just looking at generating a bunch of sequences and then comparing those to some held out test sets and looking at the kind of edit distance of the generated sequences to the test sequences.
Yeah.
Right. And using that as some sort of way to show that we're not overfitting or something like that. We have a similar distribution of generated sequences as we do of test sequences. And actually, we found that the generated sequences were a bit further away on average in edit distance from the test sequences. So they were, they were definitely not overfitting.
I see. So if you looked at like the number of subsequences of a certain size that you can match, or I guess the edit distance thing is a little bit different. But if you could get like really long matches to the training set and you couldn't get long matches to the test set, that would be an indicator that it's overfitting.
Absolutely.
Yeah.
We were also concerned about maybe it was just generating, like maybe there was one mode to the generated sequences.
Yeah.
And so another test we did, which is maybe just a naive test, a simple thing to do, a sanity check, was to actually just take the generated sequences and zip them up as a zip file and see how much it was compressed relative to doing the same thing on the test data. And we found that the compression was pretty comparable.
Oh, wait, what would that test tell you?
So it's just telling you if, so you can have, quality sequences that are generated, but maybe it's not generating a lot of variety.
Oh, I see what you're saying. Yeah. Like so the drop. Yeah.
Yeah, so we just basically take a whole batch of sequences and try to compress them. And if they're all the same sequence, you would expect that you should be able to compress it a lot easier than if there's some diversity there.
That makes sense. For a researcher who's doing basic algorithms work, do you think that DNA is going to be important for them? And do you think that there are going to be any ideas that come out of DNA research with deep learning that will impact the rest of deep learning? Just to give like one example of this from another field, a lot of the progress in attention mechanisms and RNNs has come from working with neural machine translation.
Mm-hmm.
Just because it kind of forces that to the center.
Yeah, I would say that for sure this would be the case. So it's another application area that's opening up. I think a lot of people are recognizing that biology data, health data is kind of this area where we know there's all sorts of statistical correlations, but we don't have very good models for them.
Yeah.
And I think a lot of people kind of envision it as the next big vista to explore for deep learning. So I would say, I think there's definitely, it's worthwhile exploring this area with modern tools. The question of whether that feeds back on the models themselves. I mean, I don't know for sure if it's going to happen, but for sure you might see certain ideas that come up in study of DNA that end up being kind of backported to the algorithms or backported to the algorithms. Maybe if we're studying natural language processing, maybe working on this kind of language of DNA might be a little bit simpler way to learn how to structure algorithms, and then we can move that back to NLP algorithms instead of
And then also like analytically saying what a piece of DNA does, for example, with your classifier, could be a lot more concrete than trying to say what the impact of a given generated piece of natural language will be.
Yeah, that's right. Yeah, I agree.
So for someone who's interested in working on deep learning with DNA, what do you think is a good starting point?
Well, there's an interesting paper that came out December of last year. That's probably a good starting point. But really, it's just like we were just exploring.
Wait, you're saying your paper?
Yeah, it's a bad joke.
Oh, yeah.
No, there's lots of interesting work going on with deep learning and genomics data. Our work was just one particular thing for generative models, but there's tons of work already going on with other areas, supervised learning and other things for genomics. So there's in the last few years definitely there's been a literature starting to build up. But also I would say just don't be afraid to explore. Like if you have an idea, try it out. That's the great thing about deep learning right now is it's very easy to prototype an idea, test it out, and fail fast if it doesn't work. But if it does work, then you have the tools to keep iterating on it and get towards maybe some interesting biological insights. So that's what we did. We were just doing exploratory work, right? So I encourage everyone else to follow the same kind of path.
Yeah, that sounds great. So that's the end of the questions I have. Is there anything else in particular you want to say?
No, I think that was lots of interesting questions. It was great. Thanks, Alex.
Great. Have a good day.
Thanks. Bye.
Yep.